All right, so this is the second of two lectures for Chapter 14 in Unit 3, and in this lecture we're going to talk about the electron transport chain. So, as we've mentioned before, electrons carried by NADH and FADH2 from the citric acid cycle are oxidized by the electron transport chain. Now, you may remember from your chemistry days, redox reactions or oxidation reactions. Just to review, oxidation equals taking electrons away, and reducing equals adding electrons. So the electron transport chain, as we say here, it's a system of carriers in the inner membrane of the mitochondria. And then cytochromes are proteins that contain a heme group tightly bound to the protein. And they alternate between the oxidized iron state with Fe3+ and the reduced iron state of Fe2+ as they function in the electron transport chain. So this is how they pick up and transfer and carry electrons, is via these cytochromes being oxidized and reduced, and that's how they transfer the electrons along. Complex 1, as we show here, it transfers electrons from NADH, so this NADH here that came from the citric acid cycle. And so they transfer these to coenzyme Q here. But first what they're going to do is they're going to get to coenzyme Q, transfer from NADH to flavin mononucleotide, or FMN. Then they're transferred via a series of iron sulfur centers, so FES. Apologize, this should be a lowercase e, so iron sulfur. And then these guys, these iron sulfur centers, are going to transfer to coenzyme Q. During the transfer of electrons to coenzyme Q, protons are pumped from the mitochondrial matrix here, across the inner membrane, which is here where the complexes are embedded, into the intermembrane space. So this is one of those proteins that's contributing to generating this hydrogen gradient across the intermembrane. Coenzyme Q is an electron acceptor that transfers proteins across the intermembrane in a complex process called the Q cycle, hence the name. The Q cycle, we're not going to go into the details of that just because it's a very low yield concept, even for a biochemistry exam, and especially for a board exam. So really just what you want to know is that it takes part in transferring electrons from complex 1 and complex 2, receiving those and then transferring them on to complex 3. So complex 2 receives electrons from FADH2, which again came from the citric acid cycle, which was produced during that conversion of succinate to fumarate. Now there's two places in the text where we say cytochrome Q. You should cross that out and put coenzyme Q. So FADH2 transfers electrons to coenzyme Q via iron sulfur centers of complex 2. So in here, the electrons get transferred to these iron sulfur centers, which then transfer to coenzyme Q here. All right, so now that you've transferred electrons from complex 1 to coenzyme Q and complex 2 to coenzyme Q, now you have complex 3, which is going to catalyze the transfer of electrons from coenzyme Q to cytochrome C. So you're going to have the electrons transferred into complex 3 onto one of those cytochromes, cytochrome C specifically. As a result of this, you're going to translocate protons across complex 3 into the intermembrane space. Cytochrome C is a mobile electron carrier that travels through the inner membrane, as we show here. You can see it's traveling through the membrane here, and then it transfers the electrons to complex 4. So again, to review, you have electrons coming from NADH and FADH2. NADH, they go through complex 1 to coenzyme Q. FADH2, they go through complex 2 to then coenzyme Q. Coenzyme Q transfers through complex 3, eventually to cytochrome C. And then complex 4 transfers electrons from cytochrome C to oxygen as the final electron acceptor. And this forms water. And then in the process, this powers the translocation of protons across the inner membrane into the intermembrane space here. And so here's the reaction here. Half an O2 plus 2H plus gives you H2O. And again, so you can see that all of these are contributing to creating this significant electrochemical gradient that's going to eventually power ATP synthase. So ATP synthase, it's also known as complex 5, as we show here. And then ATP synthase, which is also known as complex 5, this is the grand finale here. It catalyzes the synthesis of ATP. And this is driven by, so you've been building this up, building up all this hydrogen concentration here. and they're going to travel back down their gradient across ATP synthase back into the mitochondrial matrix, and this is going to power ATP synthase to synthesize ATP from ADP and inorganic phosphate. So again, one last time, NADH to complex 1, then it makes its way over to coenzyme Q, FADH2 through complex 2, coenzyme Q, through complex 3 to cytochrome C, and then through complex 4 up to O2, you form water here. All along, you're creating this hydrogen gradient, comes down and drives ATP synthesis. Now, again, we mentioned this earlier when we talked about products of the citric acid cycle. It depends on the source. Some say 2.5 ATP per NADH. Others will say 3 ATP. Some sources say 1.5 for FADH2. Some will say 2 ATP. Depends on the source. And as far as this goes, we already talked about the products of the citric acid cycle and then the ATP yield from those. So again, these numbers were incorrect. Just go off of the notes and the discussion we went over earlier in the lecture a few slides ago. All right, so now that we've gone through the citric acid cycle and the electron transport chain, we're going to go over some clinical pearls relevant to both of these processes. So first we'll talk about a metabolic disorder called pyruvate dehydrogenase deficiency. So if we look over here, this whole part here is glycolysis. This is happening in the cytosol. This is the mitochondria here. And then this is the citric acid cycle. So glycolysis results in pyruvate, right? Remember, it can go to lactate, which should be anaerobic respiration, and you only get 2 ATP that was produced from glycolysis itself. You get 2 NADH as well. Or it can go to the mitochondria, where then it's converted to acetyl-CoA by pyruvate dehydrogenase complex. And so if you have a deficiency of this enzyme, you cannot convert pyruvate to acetyl-CoA in the mitochondria. That's going to cause a buildup of pyruvate. It's going to back up into the cytosol. As a result of that, this excess pyruvate is going to be converted into one of two things. It's going to then drive towards being converted into lactate. So you're going to have a significant increase in lactate. This is catalyzed by an enzyme called lactate dehydrogenase, and this actually uses up NADH. So you're losing NADH as a result of this. And then it also can be converted into alanine via alanine aminotransferase, because alanine can be converted to pyruvate and vice versa. We'll talk more about that in the gluconeogenesis lecture. The inheritance pattern is X-linked recessive, so it predominantly affects males. And really, like we've shown here, so you have glycolysis and the citric acid cycle, which are, especially in aerobic respiration, are connected in tandem here, because pyruvate is then enters the mitochondria where it's converted to acetyl-CoA and then able to enter the citric acid cycle. And in this disease, there's a disconnect between glycolysis and the citric acid cycle. And this results in severely decreased ATP production because you're not getting that NADH and FADH2 produced from the citric acid cycle that then can be used in the electron transport chain to generate a significantly higher amount of ATP. So as a result, the clinical features, since you're having an increased amount of pyruvate and you're converting it to lactate or to alanine, you're going to have increased serum levels of these. And so you're going to have what's called a lactic acidosis, because lactate or lactic acid leads to a decrease in pH. And that's not good for just even overall homeostasis. The body likes to keep the pH at around 7.4. When you start getting lower than that or higher than that, it tends to throw things off physiologically, and that's just not good. So lactic acidosis is not good on the metabolic side. And then on the functional side or physiological side, it also causes neurological dysfunction because you have significantly decreased energy output. And so that's going to affect neurological development and neurological function. It also, the muscles are high energy organs. And so you're going to develop myopathy because you're going to have muscle weakness because you just can't generate this level of energy. It essentially renders the mitochondria unable to do their job of powering cells and producing energy. And unfortunately, as a result of this, it eventually leads to premature death. Now, the way you treat this is you give patients a diet high in leucine and lysine, which are ketogenic amino acids. What that means is that they're amino acids that are broken down into acetyl-CoA. Remember, in our main diagram, acetyl-CoA is a major player in metabolism, because acetyl-CoA can go, in this instance, can go towards the citric acid cycle and go around like this. help power, get us some NADH and FADH2 for the electron transport chain, or it can be converted into ketone bodies in the liver. And remember, the ketone bodies are critical for powering the brain, because the brain really only, unless under prolonged starvation, typically only is powered by glucose or carbohydrate metabolism. Now, in these patients, you want to limit carbohydrate metabolism as much as possible, because you're going to build up lactate and alanine, and you're not going to get output from the citric acid cycle and electron transport chain. In this case, you want to limit the amount of carbohydrates they're consuming, but you still have to power the brain, and so you want to make sure that they have a ketogenic diet where they have ketogenic amino acids and then also high in fat, because fat can be converted down to acetyl-CoA, and then acetyl-CoA again can enter the citric acid cycle, or it can be used to produce ketone bodies. Like I said, you also want to avoid food that's high in carbohydrates and glucogenic amino acids. Glucogenic amino acids are going to be ones that are going to be brought into and used in gluconeogenesis, which is the production of glucose within the cell. And so you want to definitely avoid that, because again, you're trying to limit the amount of carbohydrates. So you're still trying to give them as much energy as possible by kind of indirectly giving them ketone body production to help make sure the brain is supplied, and then also trying to feed the citric acid cycle kind of in a roundabout way through these ketogenic amino acids, and then fat as well. All right, so now over the next few slides, we're going to talk about some compounds that directly inhibit the electron transport chain. And the way they do that is by directly inhibiting proteins. So these complexes, complex 1, complex 3, ATP synthase. And this causes, with the exception of ATP synthase, when it acts on complex 1 or complex 3, it decreases their ability to pump protons into the intermembrane space, thus decreasing that proton gradient, which then as a result, if this is decreased, you're not going to have as much of a driving force for the protons to travel down ATP synthase and power ATP synthesis. So as a result, it subsequently hinders ATP synthesis, which makes these toxins. So these are 4 examples that are commonly seen on exams, commonly seen on board exams, biochem exams. You may have even heard of these, especially carbon monoxide and cyanide, so you can learn a little bit more about why they are so toxic. Rotenone is a component of pesticides, blocks complex 1, so it'll come down here and block complex 1, blocking its ability to pump hydrogen into the intermembrane space. Antimycin A is used for water treatment and also is very toxic to humans. This comes in and blocks complex 3. And then carbon monoxide, which is very commonly known to be very lethal gas, and then cyanide, which is also known to be very toxic. Part of the effects of carbon monoxide are due to its binding effects on hemoglobin, which we'll talk in more detail in the heme lecture. But on the cellular level, it comes down and blocks complex 4. Same thing with cyanide, very toxic, comes down and blocks complex 4. So the key things you want to remember with these is just that they're toxins, and then you want to be able to pair which component of the electron transport chain these directly inhibit. Oligomycin is an antibiotic that directly inhibits ATP synthase, so it goes right to the source, blocks complex 5 or ATP synthase, which then severely decreases ATP production, because then it doesn't even matter what the proton gradient is. If you're directly inhibiting this, then you're directly affecting ATP synthesis. And because of that, oligomycin, it's typically not used for therapeutic purposes. It's used more in research labs for that reason, because the side effects would be potentially devastating if you used it more as in a therapeutic realm. All right, so lastly, we'll finish this lecture out by talking about uncoupling agents. And these are agents that increase the permeability of the inner membrane of the mitochondria. And so what I like to think of them as is almost poking holes in the membrane. And so what that does is it allows these protons that are being pumped from the mitochondrial matrix via these complexes in the electron transport chain, it allows these protons to leak out from the intermembrane space back into the mitochondrial matrix. And what that does is it dramatically decreases that proton gradient that drives ATP synthesis. So you have dramatically decreased ATP synthesis as a result of that. And that's why these uncoupling agents can have significant effects on energy production. And what ends up happening is that when these protons diffuse back down this gradient just through these quote unquote holes in the membrane created by these uncoupling agents, that electrochemical gradient dissipates as heat. And so it produces heat, and then it also results in increased levels of oxygen consumption as well. And in the end, you also dramatically decrease energy production because this gradient is what drives ATP synthase to synthesize ATP. And so if you lose that, dramatically decreased levels of ATP production. And so some examples of this would be an aspirin overdose. This often results in fevers, which is a clinical sign. Then you have 2,4-dinitrophenol, which is an illicit weight loss substance. So the thought is here that you uncouple the electron transport chain, you're going to increase metabolism and hopefully metabolism of fats with the end goal of weight loss. And the problem with that, though, is that you're directly affecting ATP synthesis in the mitochondria, and so you can have pretty devastating side effects as a result of that, hence why it's thought of as an illicit weight loss substance. And then you have thermogenin, which is actually a naturally occurring substance in brown fat. And brown fat is typically found in infants. And brown fat functions to generate heat. And so again, thermogenin is going to act by, essentially quote unquote poking holes in the membrane. It's going to allow these protons to then diffuse back down. And as by doing that, the electrochemical gradient will dissipate as heat. And that's what's called non-shivering thermogenesis. Non-shivering because you're not, the muscles aren't causing you to shake and generate heat that way. This is an alternative way of generating heat that doesn't involve shivering. All right, so that closes out our discussion of the electron transport chain.